Synthetic biology is an emerging field, providing solutions to agricultural, environmental, and healthcare problems through the manipulation and repurposing of biological systems. As such, the need for effective biocontainment strategies have become increasingly more apparent. The purpose of these containment strategies is to prevent the detrimental consequences associated with the accidental escape or release of non-native organisms into the environment. Current strategies rely heavily on administrative and engineering controls, as well as other regulatory strategies at the molecular level. Unfortunately, many of these strategies are complex and unsustainable. We wanted to develop a more sustainable approach to biocontainment. This led to the conception of our project titled The Development of Complementary Auxotrophic Strains as a Biocontainment Strategy for Genetically Engineered Organisms. Essentially, my team and I decided upon developing an auxotrophic co-culturing system, where two strains of yeast would be grown together in a syntrophic community. Auxotrophy refers to an organism requiring supplementation of a certain metabolite in order to survive. The co-culture system would ensure that external supplementation would not be required, making this a more long-term, maintainable, and affordable solution. The yeast strains we used were of the model organism S. cerevisiae. Each yeast strain would be overproducing the molecule the other strain was auxotrophic for, resulting in the strains being mandated to stay together in order to survive. A syntrophic community also has tighter regulations on the optimal growing conditions and minimum cell density required for cells to proliferate, meaning that the chances of survival for these yeast strains in the environment would be very low. In order to determine the overall efficacy of this biocontainment system, we first had to obtain results. Our first step was to determine the best combination of auxotrophic strains and to characterize them. Thanks to the gracious support of the Murray Lab from Harvard University, we were able to obtain a set of complementary strains. One of the strains, YMM60, was auxotrophic for tryptophan but overproduced leucine. The other strain, YMM65, was auxotrophic for leucine but overproduced tryptophan. Using a standard curve for yeast growth in leucine and tryptophan supplemented media, we were able to compare the amino acid production of normal strains and our engineered overproducing strains. The results indicated that the engineered strains produced substantially more amino acid than the normal strains, confirming overproduction. The second step was to attempt to co-culture the two strains. We tested the growth of the two strains at low, medium, and high concentrations of the amino acids. In order to distinguish the individual growth of each strain within the yeast culture, one of our yeast strains was engineered to produce M-citrine and the other M-cherry, two fluorescent proteins. This allowed us to measure their respective fluorescence, indicating the growth of each strain. At low amino acid concentration, the two strains appeared to be successfully developing a co-culture, while at medium concentration, one strain appeared to be dominating. At high concentrations, growth of both strains was greatly reduced. This indicated that the strains would only thrive in a co-culture in the event of reduced initial amino acid availability, as they would grow to need each other. Increased availability led to competition, and one strain dominating the other. Our third step was to determine the growth of our engineered yeast in environmental conditions. To ensure that this was a viable biocontainment strategy, environmental samples could not have adequate amounts of amino acids available for significant growth of the yeast. We tested both of our yeast strains in soil and water samples. There was very little yeast growth in all environmental samples, indicating that the yeast strains could not survive in the environment without external metabolite supplementation. Our results thus far have indicated a favorable outlook on using an auxotrophic co-culture system as a biocontainment strategy. In the future, we aim to further characterize the co-culture system, optimize the yeast strains to be auxotrophic for the most energy-efficient metabolites, and adapt the system to be used in other microorganism species. All of the aforementioned work conducted was done by myself and my colleagues Arshia Mostelfi and Namaya De Silva, with guidance and support from all these wonderful people. Thank you.